Welcome back to the video lecturing of the sensors and measurements subject. So in this we are going to see about the biosensors. So biosensors is nothing but the smart sensor. We are going to connect the smart sensors bridging between the biological and the technology. So in this, you are going to see about the introductions. So what is biosensors, which is going to combine the biological regulation element into the physical, that means electrical signals. So this biological responses, that is the input like this enzyme, antibody or cell. So these are the physical detector. You are going to detect this physical phenomena, that is physical input that is used by the transducers into the measurable signals. So the main target is you are going to analyze the glucose toxins pathogens. So this interaction between these biological element into the electrical or optical or thermal signal. So here the signal amplifies the input data or process the data, which is going to display the readable quantity, that means a user-friendly format. So the main application of this biosensor mainly used for the healthcare application, food safety, environmental monitoring, uh, industrial bioprocesses. So the main importance of this biosensor, we are going to connect the bridge between the biological and the technology. So here going to enabling or rapid specific and into the real-time monitoring, right? So next one is the advantages and the disadvantages. The main advantages and disadvantages of the biosensors. So first we are going to see about the advantages. In these advantages, it is a mainly it is the high specificity and a rapid detection, real-time monitoring, portability, cost effective. So these all things that is advantages. We are going to, that is enzyme substrate, so the result compared with the laboratory things, portability, so monitoring, that is a continuous tracking biological signals. So all these things are mainly possible to interact or especially with the IoT integration. And you are going to reduce the need of for the expensive laboratory component or trained personnel. So the limitations of the biosensors. So the main limitations is stability issues, calibration requirements, short shelf life, high cost of the advanced devices, and the limited range. So this stability issues is nothing but the biological components, that means enzymes, antibodies. These are the things can degrade with the temperature or a pH value or the time constraints. Next one is a limited shelf life. It is going to, that means many biosensors lose activity after the limited use or use of the short range. Next one is a calibration requirements, where frequent recalibration is needed, maintain the accuracy. Next one is a high cost. So while the basic biosensors are affordable, but then the advanced ones like ISFET or IMFET can be very expensive. Some some biosensors working only within the narrow concentration range of analytics. So next one is the components of the biosensors. So the components of the biosensors is nothing but mainly biological element, transducer, signal processor, display or unit. So biological element is nothing but the input. The inputs are the enzymes, antibodies, microorganisms or living cells, which is going to specify the to the biosensor. Transducer is the converting element. So this all the a biological element is converted to the electrical things by using the electronics. Next is a processor. Processor is nothing but you have to go into strengthening or amplifies the process the raw signal into the uh, uh, readable data. Next one is a display or the final result will be present in the user-friendly format like the digital readout or on a glucometer or smartphone app or integration, smartphone app integration or display unit. So the next one is a classification by transduction type. So the transduction type is a electrochemical biosensor, optical biosensor, acoustic biosensor, colorimetric biosensors used here. So electrochemical biosensors used to measure the electrical signals such as current, voltage, or impedance. So widely used in the glucose monitoring or other medical diagnosis. Next one is some optical biosensors. Optical biosensors is nothing to detect the changes in light absorption, fluorescence, or refractions. So it is commonly usually used in the DNA analysis and the pathogen directions. Acoustic biosensors use the sound waves or piezoelectric crystals to sense the mass changes, which is helpful to the detecting the biomolecule interactions. Next is the calorimetric biosensors. So mostly that is measuring heat changes during the biochemical reactions, useful for enzyme activity monitoring. The next one is the classification of biological element. So biological element is nothing but the enzyme sensors, immunosensors, cell-based sensors. Enzyme sensors is nothing but using the enzyme as a biological recognition element, detect the substrates or the products of enzyme reactions. For example, you can take the glucose oxidase enzyme into the glucose, blood glucose sensors. Immunosensors is the pathogen detection, which is the which is going to employ the antibodies to detect the specific antigens. So highly, it is mainly used for disease diagnosis purpose. The next one is the cell-based sensors, which is used to living cells or tissues to sense the toxins, pollutants, and metabolites, which is going to provide the bacterial sense sensors, which is monitoring the environmental things only. Next one is advanced biosensors. Nowadays, all the biosensors which is used in the advanced one. So the first one is a non-sensitive field effect transducers, immunologically sensitive effect, and blood glucose sensors. So the non-ISFET is which is mainly used to detect the specific ions like H plus, which is used in the field effect transducer. Look at the picture here. So this picture we can provide like the field effect transducers, which is provide the rapidly miniaturized, that is a highly sensitive measurement. So that is a commonly used in the pH sensing and medical diagnosis. Next one is the immunologically sensitive factor, which is used in antibody recognition with the PET technology. So which is a highly specific direction of antigens. So it is also used for disease diagnosis. Next one is a blood glucose sensors, mostly widely used biosensors in the healthcare device. So which is going to employ the glucose oxidase enzymes with electrochemical transduction. So it is used to mainly used portability and user friendly also. It is widely integrated with the IoT. We can connect for the real time monitoring. So next one is a sensors integration in IoT. So the main concept of this IoT is nothing but we are going to link with the IoT, links with the biosensor, that is to connect the digital network for real-time data collection, analysis, and monitoring. So the main concept. So each biosensors act as a smart node and transmitting biological data to the cloud platforms or mobile devices. So the main things of this real-time health monitoring is nothing but a biosensor directly biological signals, that is glucose level, heart rate level, this in the alarm. So we are widely used, and also we can connect with the wireless modules like Bluetooth, Wi-Fi, Zigbee. We can send the data to the cloud, and correspondingly we can monitor in the disease diagnosed by the uh, clinicals. Okay, so the main advantages this sensor integration with the IoT, you can enable the real-time health monitoring, and also you can remote diagnosis and telemedicine, and also provide the predictive analytics things. So that means we can use it for early disease detection. And the next one is the you can integrate with the smartphones and the cloud storage for the easy for access. So the main application is nothing but wearable health trackers. So which is used in the oxygen sensors, that is heart rate level, which is continuously monitoring, and also you can use the smart glucose monitoring for the diabetic patients.